I'm gonna beat a dead horse, just in case.
Now it's the end. Oh, no, I know. I don't know how I
So
did
let's
that.
say that
Oh, perfect. Okay.
this is our enzyme, our Pac-Man. This is PFK, phosphofructokinase. Okay. What is the substrate phosphofructokinase converts? This guy? Right? So we could say that is the shape of, we'll call it F6, fructose 6 phosphate. So normally PFK would act on fructose 6 phosphate. It would add a phosphate group to that. And so the reaction that goes forward would result in the production of, I don't know, a square, which would be F16PP. I don't know, I'm making that up. Fructose 1,6-bisphosphate. So enzyme takes a reactant into a product. Clear? Now, this enzyme is allosterically regulated by phosphoenolpyruvate, PEP. Let's say it's shaped like a circle. And so the PEP comes in and it binds at the allosteric site on this enzyme PFK. It's got a spot where it binds there. And now that it has bound to that allosteric site, suddenly the active site of PFK looks different. And it can no longer bind its substrate to convert it into product. How we doing?
Good.
Good?
Yeah.
Good. Got one good, a couple giggles. Okay. We can always revisit this. Let me know if this becomes confusing to you. I don't think there's a question on your assignment that can cover this adequately, but it will be on your exam. So, if you don't understand this, if you cannot explain it in an essay question.
It's just the feedback cycle for pyruvate,
Yep,
right?
Yep, it's
Okay, yeah.
feedback inhibition for the production of pyruvate.
Right.
Okay. Who is the allosteric regulator or the allosteric inhibitor? PEP. What enzyme does it inhibit?
PFK.
Yeah.
F6B.
PFK. It inhibits the enzyme PFK. It inhibits the enzyme which converts fructose 6-phosphate into fructose 1,6-bisphosphate. So it prevents this entire reaction from happening.
Are those substrates?
This is a substrate, this is a product, which is then used as a substrate to make another product. So it's a series of chemical reactions. So on day one, we said A to B to C to D, whatever. A, B, C. So it's just this entire process of metabolic reactions. Okay, so we will practice this in class. We will practice this on the day that we review for the exam on Thursday. We'll go over a question that kind of covers all of this. But if you have any questions or you feel like this is not clear, please see me and we can talk about it. The reason that I think this is so important is because it is like an angel of an example for allosteric inhibition.
Okay.
One last question. Would you call this competitive inhibition or non-competitive inhibition?
Non-competitive.
I got one competitive, one non-competitive. This is non-competitive inhibition because the inhibitor, first of all, looks nothing like what would normally bind the active site. So it's not competing for the active site to block the enzyme in that way. It's playing dirty. It's changing the game entirely by binding to that enzyme and changing the site completely. Okay. I think that would be glycolysis, guys. Any questions? You're doing great. So again, anything in red is what I want you guys to know. Okay, so then from there, pyruvate has a lot of different fates. So pyruvate, the product of glycolysis, is an awesome metabolic participant. It has all different kinds of pathways that it can then take to make different kinds of products. So in the case of aerobic respiration or anaerobic respiration, we're going to just follow that right into the citric acid cycle and onto the electron transport chain. But pyruvate can also be used in anabolic reactions or synthesis reactions where we're making a product. So we can use pyruvate as a starter molecule or an ingredient to make amino acids, sugars, fats, etc. So anything we want to build, we can use pyruvate for that. So it's a really, really useful molecule. And if we can't carry out the Krebs cycle, there's always fermentation, right? So pyruvate is what's also going to be fed into fermentation, which is one of our favorite metabolic pathways, because if you eat yogurt, if you drink wine, anything like that is all going to be thanks to fermentation. So we're going to follow the fate of pyruvate to respiration at this point. But first, remember, pyruvate itself is not what enters the TCA cycle. So it's got to be modified before it can be sent into the TCA cycle. So it doesn't look exactly like pyruvate. Instead, it's modified. And this is where we talked about another carrier molecule, coenzyme A. In that video that we watched, we talked about coenzyme A, right? So pyruvate is going to be converted into an acetyl group, which is just chemical jargon. You don't have to know much about that. And then that acetyl group is going to be attached to coenzyme A, and coenzyme A is going to carry the acetyl into the citric acid cycle. So it's like it's catching a taxi to the TCA, okay? In this process, all that I want you to take out of this is that, one, pyruvate is not what goes directly into the TCA cycle, and two, we get some CO2 out of the reaction. So CO2 is a product of this, which explains the CO2 that we breathe out, part of it. And we get some electron carriers picking up electrons as well. So NAD+, one of our favorite pizza delivery guys, is picking up electrons from this reaction, and it's going to deliver them to the electron transport chain. So this is just the transition step. One, you do not by any means have to memorize this chemical equation. Just know pyruvate itself doesn't go directly in. Now we have the Krebs cycle. We're going to cover even less of the Krebs cycle. So there are only a couple of things that I want you to take out of the Krebs cycle. One is that we are taking, at this point, 2 molecules of acetyl-CoA. So maybe we should go back to glycolysis for just 1 second and look at the product. So we start out with 1 molecule of glucose. We end with 2 molecules of pyruvate because we've taken 1 6-carbon molecule and split it into 2 3-carbon molecules. So from here on out, everything is going to be multiplied by 2 because now we're working with 2 molecules that are being fed into everything else, okay? So that's important to know. So 2 pyruvates are going to be made into 2 acetyl-CoAs. Then those 2 acetyl-CoAs are going to be fed into the citric acid cycle, 1 at a time. So for every glucose molecule, how many times does the TCA cycle move forward? 2. So it cycles twice. TCA cycle and Krebs cycle are interchangeable words. So whatever you have learned previously or whatever you can
And you
identify
just said
with.
citric cycle also.
Citric acid. TCA is the citric acid cycle.
What's the T again?
The
Okay.
citric acid. Sorry. Yeah. I didn't mean that in a, sorry.
That's
The
cool.
citric acid.
The Krebs cycle and what else is the same thing you said?
TCA,
Okay.
citric acid, or Krebs. Yeah. Sorry. Lots of words. I imagine, does anyone know why it's called Krebs? I actually don't. Probably the guy.
Yeah.
Unlikely a female, probably the guy. Okay. So, here we have our 2 pyruvic acids from glycolysis being converted into acetyl-CoA in the transition step. And then the acetyl-CoA immediately is converted into citrate or citric acid. That's why we call this citric acid cycle, because the first product that comes out is citric acid. And then basically from here, what happens is we start breaking down that citric acid into different molecules. So just a bunch of reduction and oxidation reactions. So as this citric acid is converted into different molecules all the way throughout this cycle, at some point electrons are going to be released and we're going to lose carbons because we're breaking things down and taking carbons off and stripping them off. Every time we strip off a carbon, it's going to leave in the form of CO2. So again, that CO2 that we breathe out through aerobic respiration is coming from here as well as the transition stuff. So we get some CO2s coming out of here. And we also get our NAD pluses, which are our electron carriers, picking up electrons to be delivered to the electron transport chain. So you don't need to memorize at what point those steps are releasing CO2 or are taking electrons to the electron transport chain. You just need to know that that's a huge part of what's happening in the citric acid cycle. Okay? In this entire process, we are going to make one ATP. One measly ATP the whole time. Luckily, it doesn't cost any ATP, so that's good. We know we're making a good investment. So you can see that happening right here. Another step that is important is this new electron carrier that's been introduced, which is FAD. So from the process of succinate, some chemical, being converted into fumarate, some other chemical, electrons are picked up from a new electron carrier, FAD. FAD, as it picks up those electrons, is going to be converted into FADH2. So that's what you see happening here. Can anybody tell me something interesting about this FAD being converted into FADH2, if you did your reading? Your book totally skims over it, and I think it's so cool, but your book doesn't. This is actually right here attached to the electron transport chain. They're like neighbors. So the electron transport chain is a bunch of complexes, right? If you looked at the pictures, it's like a bunch of huge protein molecules. This process right here is complex 2 of the electron transport chain. So actually the citric acid cycle is directly attached to the electron transport chain in this case. So the electrons that are coming out of here at this point are directly attached to the ETC. They don't have to travel very far at all. They're in the same office, different cubicles. Okay, that is pretty much all I want you to take out of the Krebs cycle. One is that we get 2 ATPs being made. So I said 1, and I'm not wrong, but 1 for every 3 carbon molecule that comes in, right? So it's going to move twice per glucose. So the math, 2 ATPs per glucose are being made, okay? We get 6 NADHs. That's way more than what we saw being made in glycolysis. So lots and lots of electrons being picked up. And 2 FADH2s also being made. So again, electrons being picked up. So we have 8 total electrons being picked up here. So if I were to give the Krebs cycle a middle name, I would call it Krebs reducing power cycle. And the reason for that is because we are gaining a ton of electrons in the form of reducing power. So gain electrons, reduce. Lots of things are being reduced here. So that is the major product of the Krebs cycle, is these guys being reduced. Very important. So huge contributor to the electron transport chain. So it's okay that we only make 2 measly ATPs in this process, because it's going to pay off later in PTC. Any questions about this? That's all I want you to know. What's up?
That's exactly what our tutor said. She will never let you know the numbers of what is.
That is not true. Your tutor is one.
My first biology teacher made us memorize
Oh yeah,
everything.
I had to memorize this in my micro class, and I made one of my classes memorize it. But guess how well they memorized it? Not very well. So that didn't go well.
Do we need to know the locations of where these happen?
Where these are happening?
Yeah.
So what I want you to pay attention to is that it's going to be different in prokaryotes than eukaryotes. So in prokaryotes, there is no mitochondria. Most of this is happening in the mitochondria of eukaryotic species, but prokaryotes don't have one, so they're doing it somewhere else. And so we'll talk more specifically about locations when we get to the ETC, but for this stuff, don't pay too much attention. Good question. Okay, here we go. We're almost there. So the electron transport chain. This is where we get the bulk of our ATP being synthesized. So everybody else is like B team at making ATP. This is the A team. This is where we're making tons and tons of ATP all at once. And two ways to make ATP. Oxidative phosphorylation, that's what's happening here. What's the other way?
ATP synthesis.
The substrate level phosphorylation, which is happening in glycolysis and the Krebs cycle. We didn't really go over the process in the Krebs cycle, but it's happening there too. Okay, so what's going on in the electron transport chain? This is, I like to picture this as a relay race of electrons. So whereas normally you would hand off a baton in a relay race, you're handing off electrons here. So it's just a chain of a bunch of proteins that are receiving and handing off electrons from electron carrier molecules. Okay. Depending on who the electron carrier is, it's going to release its electrons at a different site. So the NADH that is coming from glycolysis and from the TCA cycle, those guys are going to release their electrons at complex 1, and we'll look at a picture of that. That's where they're going to drop their electrons off at. The other station where electrons can be dropped off at is complex 2. FADH2 is complex 2, so it is not having to travel very far. It's just handing them off from there. Okay, and then the electrons are just handed off from there on down from one carrier to the other. And the last one to pick up those electrons is ATP synthase, which is an enzyme. So anytime you hear A's, kinase, isomerase, ATP synthase, those are enzymes, okay? ATP synthase is a monster of an enzyme. It is bigger than polymerase. It's huge. And ATP synthase is a machine that makes ATP. So synthase, synthesis. It is a synthesis machine. It makes ATP, okay? And so it is going to carry forward that reaction that makes ATP, which is ADP, diphosphate into triphosphate. Hopefully by now you should know that reaction. Now, what powers the ATP synthase machine, because it definitely requires power, is what we call the proton motive force. So we will not talk about neutrons in this class, but we have protons, neutrons, and electrons, right? Protons are what charge? Positive. Electrons are negative, right? So the proton motive force is the positive charge that is powering the ATP synthase. And we'll see how that works here in just a minute. So this is going to result in 34 total ATPs per glucose. So that's a lot compared to a measly 2 here and 2 there. This is really good at making ATP. So let's watch a video of how this happens. Here's a picture if you want to follow along with your video. These are protons, positive charge. You can see our negatives are hanging out down here. So just keep that in mind as we watch this video.
Glucose is oxidized.
Sorry.
And the Krebs cycle. The coenzymes NAD+ and FAD are reduced.
I'm gonna restart that. Eukaryote. Mitochondria.
When glucose is oxidized during glycolysis and the Krebs cycle, The coenzymes NAD plus and FAD are reduced to NADH plus H plus and FADH2. In the mitochondria, the electrons from NADH plus H plus are transferred to the electron carrier proteins, and the protons are transferred across the membrane. As the electrons move from cytochrome to cytochrome down the electron transport chain, more protons are carried across the membrane. Cytochrome C transfers electrons to the cytochrome C oxidase complex. Protons are also transferred to the outside of the membrane by the cytochrome C oxidase complex. The cytochrome oxidase complex then transfers electrons from cytochrome C to oxygen, the terminal electron acceptor, and water is formed as the product. The transfer of protons generates a proton motive force across the membrane of the mitochondrion. Since membranes are impermeable to ions, the protons that re-enter the matrix pass through special proton channel proteins called ATP synthase. The energy derived from the movement of these protons is used to synthesize ATP from ADP and phosphate. Formation of ATP by this mechanism is referred to as oxidative phosphorylation.
Okay, so this gets pretty complex. I do not want you to memorize the names that video used. That's why I didn't really talk about them much here. What I want you to know is they go in order, 1, 2, 3, 4. And electrons are basically being handed off from one complex to another by electron carriers. So what you'll notice is that the electrons that are delivered from FADH2, right there at complex 2. So they skip complex 1. They're one step ahead, the ones that are delivered from FADH2. So they don't have to go through complex 1 at all. And so that being said, the electrons that are fed into complex 1 that are dropped off by NADH don't have to go through complex 2 either. They skip over complex 2 and hop to complex 3. Okay? And then they all kind of convene at complex 4, which is where oxygen is going to accept those electrons and become H2O. That's what we see happening here. We have our oxygen having electrons accepted and becoming H2O. So that's a product. The other major product besides H2O is going to be ATP, right? So here is where we see that big machine ATP synthase. ATP synthase does not just operate on its own. It's not battery powered. It's powered by the proton motive force. So every single time electrons are passed off, they have corresponding protons. For every electron, there's a proton. That's just the way chemistry works. You don't have to think about that in too complicated of a manner, but for every electron, there are protons pumped. So every time we see electrons being passed off, protons are pumped up here. And so now we get a gradient, right? We have a bunch of positive charges up here and negative charge down here, which is actually very similar to a battery. So the positive charges are the positively charged protons, they don't have a way to get back in here. They can't just make their way through the membrane. As you guys know, that membrane acts as a bouncer and says, no thank you. And so the protons instead are going to trickle through here, through the ATP synthase machine. And they are going to act as what powers that machine. As the machine moves, it brings together ADPs and phosphates and Hulk smashes them, creating an ATP. Any questions about this? So there's a couple, if we go back to this slide, there's a couple words on there.
Right.
Chemiosmosis, that's that proton gradient. That is that idea that we have positive charges and negative charges. So there's a chemical gradient. So we call it chemiosmosis. The protons are what power ATP synthase. So we call it the proton motive force. Okay. Are we clear? Any questions about aerobic respiration? Okay, let's leave off there. And we'll pick this back up tomorrow.